And we're not going to look at this slide first. Even though we're looking at this slide first, we're not looking at what this slide is about first. One more important thing about meiosis is just understand the basics of the process. That's what we're going to deal with, is just in a nutshell, without any weirdness whatsoever, what do we do during meiosis? But what really makes meiosis fun and interesting and difficult is the fact that there is genetic diversity that is introduced during the process. And we will spend most of our time on this particular process here. We call it crossing over. It's a recombination process where DNA from one chromosome gets swapped with DNA from another chromosome. Then we have the process of independent alignment, which is going on during metaphase 1. Again, we're not going to really deal with this right now, so we'll come back to this in a second, or a minute, or several minutes. But from a holistic view, there is a symmetry to the process of meiosis. First, again, we have meiosis 1 and meiosis 2. What is important to remember is we separate different DNA molecules from each other during the first and second meiosis. And in particular, we want to remember it is the homologous chromosomes that are going to be separated during the first meiosis. So our maternal chromosome number 1 will end up in a different cell than our paternal chromosome number 1. Same is true for chromosome number 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 22. And different X's go to different cells, and one X and one Y, depending if you're male or female. We're going to separate the homologs from each other during the first meiotic process. And during our second meiosis, we will deal with the separation of the sister chromatid pairs. And just to remind you, every cell division starts with DNA duplication. Both meiosis and mitosis, we go through a process where the cell is diploid, and then it goes to a place where it's still diploid, but now has twice as much DNA that's present. So we're going to copy our DNA before we start anything, and that's something we always want to remember. Now, as I was looking at these slides just a few minutes ago, I was thinking, maybe I should use a different word than prepare, but hopefully you notice the difference between what is stated for prophase 1 and 2 and metaphase 1 and metaphase 2, because they may spot the difference.
DNA and 4 switched.
Yeah, so first we're preparing the DNA, then we're preparing 4. Now I was going to walk over here and think maybe I should use a different word like ready. Which is kind of the same thing, but prophase 1 and prophase 2 are about getting the DNA ready for separation. It's not moving anywhere yet, but we're doing everything we need to do to be able to move it. All the movement that's going to go on during meiosis, both 1 and 2, and during mitosis, is going to happen during the metaphase and anaphase cycles, or phases. Prophase 1 and, at least prophase 1 and 2, and telophase 1 and 2 can be thought about as being opposites. We get the DNA ready at the beginning, we do the opposite at the end. Imagine this is a trip. First thing you do is you pack your luggage. That's the prophase idea. Packing things up, getting things ready. Then you drive to the airport, and then you actually go somewhere long distance way. And you come back. So metaphase is going somewhere, anaphase is coming back from somewhere. And when you're home, you unpack. So think about telophase and prophase as being opposites. We're getting the DNA ready to move, but after we've already moved, we have no more need for moving the DNA. So we do everything in reverse. But where a lot of those sort of fun stuff happens is in the metaphase and the anaphase. And you can see from meiosis 1 versus meiosis 2, a lot of we separated from each other. Just to remind you, homologous chromosomes during the first meiosis and sister chromatids during the second meiosis. Again, this is just a bare bones idea of what happens during each of these different phases. Prophase 1 is where we're going to see the most difference. There's a lot of extra things that go on during prophase 1 that don't go on during prophase 2. Metaphase 1 is also a place that we'll see some differences or divergence from metaphase 2. Now, I hope that makes sense. There's a genetic diversity event that happens during prophase 1 that won't happen during prophase 2. There's a genetic diversity event that happens during metaphase 1 that doesn't happen during metaphase 2. Well, we make the DNA ready to be separated, and during metaphase, what we're going to see is all the DNA goes to the center of the cell. We call the cell midpoint, which is where the word meta comes from, meaning middle. We pull all the DNA in the middle, then we pull all the DNA away from the middle. Anaphase is the separation of the DNA. We're going to see a diagram of this in just a second. And then telophase is just, let's turn everything back to what things were at the very beginning. So just keeping this in mind, we pair the DNA, we move the DNA to the middle, we separate the DNA, and then we go back to sort of the default state. And then we do it again for the second round of meiosis. So here's just a basic idea of what we do, just to save on space, so we don't write phase after pro. Hopefully you know that pro means prophase. There's also not a Roman numeral there because it says meiosis 1. How many chromosomes does this cell have at the beginning of prophase? 6 is the right answer. So they may disagree, or they may not understand why it's 6. Nobody has any questions? They say, oh yes, 6, logically the right answer. Don't ever think about it whatsoever. I'm never going to get this wrong because I completely understand this principle and process completely. 100% completely. Everybody feels that, yeah, easy. Simple stuff, right? Only one person wants to admit it, but not too much, just a little bit. Admit this problem. Without really knowing anything, it's hard to kind of decide, but think about what we do before we start any cell division. That is sort of a beginning point. First thing we have to understand when we think about number of chromosomes is they're always going to be diploid. We have to have an even number of chromosomes, which means we have to have an odd number of, or at least could be an odd number or even number of pairs. And so off the bat, it gives us an idea of how big is this thing would be. Remember, diploid means two end. That's the other thing. So we always have the diploid cell to start with because we haven't separated any DNA or moved things around. Also remember that when we start prophase, we have already duplicated DNA. Which means we are now in what we call this 2 to the 2n state. Each chromosome is now a sister chromatin pair. So now we just need to count up how many pairs of chromosomes are there. And generally speaking, when we draw sister chromatin pairs, we always kind of use the same format, even though it's not really correct for how they look when we're actually viewing them microscopically. But for ease of looking at things, every time we draw this little X pattern of DNA, that means this is a sister chromatin pair. So 1 pair, second pair, 3, 4, 5, 6 sister chromatin pairs, meaning 6 total chromosomes. So our diploid number is 3. Now you can also guess it by the colors. Black, red, and purple representing different number of chromosomes, potentially. So we have 6 total chromosomes in this cell, 3 pairs of homologous chromosomes. Each chromosome is a sister chromatin pair. Really important to make sure we understand that basic starting point. We have 6 total chromosomes, but there's 12 pieces of DNA. We need to do something to separate the DNA from itself and then from itself as we go along. Does anybody have any follow-up questions to how do we sort of look at how much DNA is present inside of the cell? Typically, there's a genotype associated with it already, but I didn't show it to you right here because you can see it sort of later on. But this genotype is heterozygous for 3 different chromosomes. It'd be capital A, lowercase a, capital B, lowercase b, capital B, lowercase e. Because it's a heterozygous for 3 different genes, A, B, and E. Why don't we use letter C? Or why don't I use letter C?
Because they look the same.
Yeah, because the capital and lowercase look the same. So I try to use letters that don't look the same. Capital and lowercase, because that is just annoying to have to deal with. But in prophase, we're doing lots of things you can't see. We're getting the DNA ready to be moved, and then when we move to metaphase, all the DNA gets moved to the middle. Now the orientation in the picture here, we'll talk more about as we go forward, but basically you put your A chromosome set somewhere. It could be in top or the bottom, doesn't really matter where the A is, relatively speaking, but it's gonna line up and move to the middle. B is gonna move to the middle, E is gonna move to the middle. And once it's in the middle, doesn't stay there very long, and we move to anaphase very quickly. When we look at meiosis and mitosis, we can take a snapshot of the, like pause the process, say this is what sort of typifies metaphase. But what the cell is actually doing is non-stop process, and something you should keep in mind. That DNA moves to the middle very quickly and then separates very quickly. It's like snap apart. There's very little time for the cell to sit there and say, am I in the middle? Yes, no, maybe. But generally speaking, try to make sure everything's in the middle. Then we separate it. I'll talk a little more about this later. If anybody think about a reason why it would be useful to move everything in the middle before we pull everything apart. Anybody have any thoughts on why is it important to get everything in the middle first before you pull everything apart? Why not just, separate black one from the black one, the red one from the red one, the purple one from the purple one, wherever they are to start with.
To make sure you get one of each on both sides.
Do you really need to put them in the middle just to make sure that they go to the opposite sides? Maybe. That's not the main idea that I think people typically talk about.
So they don't tangle?
I'm sorry, what?
So they don't tangle, sort of?
Not necessarily that, but you're sort of getting closer to a more appropriate answer. Did you have a thought?
I'm just thinking about organization, keep the dominant on one side and recessive on the other, so it's easier to split.
That is something that is not going to happen, because the cell doesn't know what's dominant or recessive. And that's the independent alignment idea we're going to talk about in a second. It's going to deal with whether something's dominant or recessive and how things line up. The question is, why do you go to the middle before you pull things apart? Has anybody ever had to share their Halloween candy with someone else? How do you split up the candy at the end of the night? I'm sorry, what? So how do you determine what's good and bad? You put everything in a big pile together and then say, this one's mine, that one's yours. This is mine, that one's yours. It's sort of a basic process for dividing things, right? Put it all in one pile. So you get one, I get one. You get one, I get one. And so realistically, we go to the middle so that everything moves in equal distance. You're on the right track, you're on the right track also. But to move it to the middle means everything moves the exact same distance for the exact same amount of time. And so if this is a timed process, then once everything sort of gets in the middle, then everything's going to move the same distance. If they're not in the middle, then some things have to move further than others, and it's harder to coordinate things. When some things are more using or moving a short distance, it takes a short time, and some things are moving a long distance, it takes a longer time. But also, the tangled idea is important because things are sort of going past each other. Maybe it's going to screw up the ability to separate them. They're all right in the center, they're all being pulled apart at the same time, the same distance for the same amount of time. It's an organization. Important thing is just make everything as organized as we can. So we pull them apart. Now the other thing is, why do we pull them apart? So we can cut the cell in half more efficiently. Now it's also represented here, but maybe you notice the cell does change shape a little bit. What is pulling the DNA apart is also thinning the cell. So it's a little bit easier to sort of cut it down the middle. Now even though prophase and telophase are not going to be exactly opposites, some of the things we do during prophase are going to be undone during telophase. And to make this picture really correct, we would need to have a nucleus present in prophase and a nucleus present in telophase, then disappear really quickly or reappear really quickly. But look at the genotype of these cells now. Do we have homologous chromosomes present in this side or in the other side? No, we don't. And again, one thing we want to think about is typically we identify homologous chromosomes by, does it have the dominant allele or the recessive allele? A capital A versus lowercase a. Sometimes they're going to be homozygous, both capitals or both lowercases, but at least some of them are going to be heterozygous. So we can see the movement of some things. But most of the time, they're going to be homozygous. And so in this case, just because of happenstance, all the dominants were on one side and all the recessive on the other, which means we get this picture at the end of the day. We separated the A homolog from the other A homolog, and separated the B homolog from the other B homolog, and E from E. But we still have our sister chromosome pairs. So we've separated half of our DNA, but not the whole thing. And if we're thinking back to what we talked about on Friday, these cells that are now on telophase are the 2-1N state. Because there's no longer diploid, so they're now 2-1N. They're still sister chromatids, so we call the 2-1N state. And these cells are going to go forward for the next round of meiosis. Remember, in between meiosis 1 and meiosis 2 is this idea or time that we call it interkinesis, which is just the time between the beginning of one and beginning or end of the other. I guess literally be the end of number 1 and the beginning of number 2, but regardless, these two cells are going to go forward. And just for space, only one of them is going to be shown for the next stage. And it's the one with all the capital letters. And so prophase can get the DNA ready to move. Metaphase, move all the DNA to the middle. We no longer have homologous chromosomes or 4 pieces of DNA, we only have 2. So we line up our DNA in the middle. And once it goes to the middle, we pull them apart really quickly. And as we pull the DNA apart, the cell sort of elongates, so we can cut it down the middle much more easily. And then during telophase, the DNA is in opposite cells, we reform our nucleus, and those are our gametes. You can see they're haploid. Really, truly haploid now. No longer 2 copies of A, no longer 2 copies of B, and no longer 2 copies of E. Just 1A, 1B, and 1E. Now where the differences are is what letters go together and whether they're capital or lowercase. And so we'll look at it as we go forward. Just as a basic picture, hopefully everybody can look at a basic idea of meiosis and understand that here we're separating this type of DNA molecule called homologous chromosomes, and here we're separating the sister chromatids during the second phase of meiosis. I'm never going to ask you to draw out meiosis for a test or a quiz. I don't think I'll ask you to identify phases, because there's other things we'd rather be doing, like dealing with crossing over and independent alignment. So think about metaphase and anaphase as being opposites. Move the DNA to the middle, pull the DNA apart. Prophase and telophase are going to be fairly well opposites of each other. And in particular, the 3 things that will happen during every prophase and every telophase are shown here. At least every prophase. This is also going to be true for the prophase of mitosis as well. And so the 3 things we need to do to get the DNA ready to be moved around. First, we have to get rid of the nucleus. The nucleus would keep the DNA from being moved around. It's a membrane barrier. So get rid of it. Then we're going to make the DNA smaller. We go beyond the chromatin idea, make things more compact than they were already. And if you look at a cell under a microscope on prophase, or at least before prophase, you can't see any individual chromosomes. If you look at the same cell in late prophase, you'll see individual chromosomes, because the chromosomes condense, get much bigger, much more compact, and are much more visible at that point. That's just to make the DNA able to be moved. The thing that actually moves the DNA are what we call the spindle fibers. And generally speaking, the spindle fibers are 2 basic parts. Some people just refer to the spindle fibers as the microtubules. Some people lump them together with the kinetochore, which you might pronounce kinetochore, chore. And what's a chore? What is a chore?
Something you have to do that you don't really have to.
But it's work. That's something that might be helpful. It's a kinetochore because you're moving something. And also, what does kinetic mean? Typically means something about motion. So the work to do motion, the thing to do motion, I don't know. Kinetochores are we call the movement machinery. It's a protein that actually physically moves the DNA to the middle of the cell during metaphase, and then towards the cell poles during anaphase. When we talked about chromatin, one of those histone proteins was replaced by another protein. It was H3, and we replaced it with a protein called SINPA. SINPA protein is the catalyst for calling the microtubule proteins to come here, which should make it go to the centromere. It's not exactly true to say this, but is this an epigenetic process or not epigenetic? I'm sorry?
Kinetochore assembly is epigenetic.
Why would you say that? Does it happen everywhere?
No, I mean, it does happen at the centromere, but it doesn't bind based on the DNA sequence, which is why
Right.
it's because it's epigenetic.
Which is why I said it's not exactly epigenetic, because it doesn't care about the DNA sequence. The DNA sequence around the centromere is a particular type of DNA. We call those alpha satellite sequences. So it might not actually care about the specific A, T, G's, and C's, but it does care that it's the alpha satellite DNA, which means we only replace the H3 in one location, which means all the other nucleosomes are still containing H3. So we make a force, a kinetic force, to go to one specific location. That's our anchor point. We're moving the DNA around the centromere of the chromosome. Doesn't matter if it's in the middle or in the end, it is the anchor that we use to move our DNA around. For us, we don't care about the actual mechanics of how it works. Maybe you'll deal with that in cellology, but who knows? If you want to know, I'll tell you a little more about it. We don't care. Kinetochores are the machine, the microtubules are the tracks. Choo-choo train, moving stuff around. It's an easy way to think about it. So we make our DNA smaller, get rid of the nucleus, and then attach these really long ropes and chains to our DNA so we can carry it on. Now, all these things are going to be reversed when we look at telophase. So make them don't need the kinetochores anymore, so get rid of those. So if you get rid of all the kinetochores, it means we potentially have to kick out all the SINPA proteins and pull in all the H3 proteins again. Now I will tell you that I don't know that this actually happens. Maybe during angiogenesis, the SINPAs still stay there and the H3s are still gone. I have no idea. But maybe they do disappear and get replaced and go back to normal. Maybe they don't. I have no idea. But during telophase, we should no longer need to move our DNA around. So we get rid of the kinetochores, we no longer need those microtubules. Our DNA can decondense, go back to regular chromatin, and then the nucleus would reform. Now some of these things will happen, some of them will not. It really depends on how fast is interkinesis. Especially when you go from telophase 1 to prophase 2, you don't have it all the time, and some of this you don't really do fully. You just do a little bit or don't do anything at all because you know you're going to undo it really quickly. So there's really no way to tell exactly which cells are going to perform all these functions completely. But that's the basic idea of telophase, is just reverse the things that go on during prophase. After telophase is cytokinesis, and we don't talk about cytokinesis. It's never going to show up in a slide, I don't believe. So you just need to at least be aware that cytokinesis is going to occur. It's the thing that actually separates the cells from each other. Because telophase does not separate the cells. It just reforms the nucleus so that you can then separate the cells. Again, get the DNA ready to move it to the middle, pull it to the sides or the poles of the cell, and undo all the things we did at the beginning. And do it again. There's different DNA molecules separated first compared to second. And that's pretty boring. Right? It's pretty boring. Maybe it's not boring. Who thinks it's like the most exciting thing ever? Yeah, yay! Yay for meiosis! For me, it's hard to pick a favorite phase. because my name's in every phase. So if I had to pick, I just can't. Maybe it's because I would pick prophase 1, because it doesn't really matter. It's just new and fun and exciting things that happen during prophase 1. These are things that will not happen during prophase 2. It's important to state that. Also, these are things that will not happen during the prophase of mitosis. These are things that are strictly unique to the meiosis prophase 1. Synapsis has to happen first, and then crossing over can happen second. Synapsis is an important process that will happen all the time. Crossing over is a rare event, so it doesn't always happen. But generally speaking, crossing over will happen at least 23 times. One for every chromosome, because we generally say that every chromosome encounters at least one, or undergoes one crossing over event every round of meiosis. But generally, one thing that will be really important going forward, the fact that synapsis is an absolute requirement for meiosis to work, but crossing over is a rare event, so it may or may not happen. Sometimes it might happen twice, 3 times, but each time you do it again, it becomes more and more rare. When we start this process, we need to kind of go backwards in time a little bit. Please, you're going to draw on board a second. One of the really important ideas of synapsis is the word that it kind of sounds like, which is snap. If you had to describe the word snap or what snap does, what would you say? You think snap breaks? Possibly. But not for us. A snap can also have another meaning.
Come on.
Come on. I know it's weird. Snap sometimes means come together, but also snap sometimes means apart. But for synapsis, and the idea of snap is when you snap things together, this is why it's called snapping together, because you're snapping things together. But we're going to see a joining go on. And the question is, what is joining? But we'll get to that in a second. And crossing over, imagine you're walking down the street and you cross over. What are you doing?
Switch sides.
Yeah, you're just switching sides. And so there's a switching that has to happen. Now again, we'll talk more specifically about what is joining together and what sides and how the switching is going on. But that's generally what we need to appreciate with synapsis and crossing over, is we're going to bring things together and then maybe switch some things. Now we go back to the very beginning. Before we even get to prophase 1, we have a cell. It'll be a diploid cell. Actually, let's just combine these together, because that'll help out for this first phase. 2 chromosomes. Each chromosome has 2 genes, gene A and gene B. And we have a heterozygous genotype. Capital A, lowercase a, capital B, lowercase b. It's a fairly simple one, but it's important to understand to be able to look at what we have. So before we do anything, first thing we do is we duplicate our DNA, so we get a sister chromosome pair of the capital A, capital B chromosome, and a sister chromosome pair of the other chromosome, the lowercase a, lowercase b. So we're our DNA duplication stage. Doesn't really work out for duplication, but 2n, where n is 1, and here we are still 2n, but now we have twice as much DNA, so we have that 2 for the 2n stage. Rid of that, but still, this is already a good picture. I'm just trying to remind you of the way we talk about things. So this is before we even get to prophase. So sometimes we talk about using some mitosis language. This is G1, this is G2, and what happens here is what we call S phase. You really don't need to memorize that until we get to mitosis, but just for hooking things together or linking things together, here's sort of where we are. And this is the thing that's going to enter our prophase. And so the first thing we have to do is to do this synapsis idea. Synapsis is defined as the homologous chromosome pairs joining together. But each homologous chromosome is a sister chromatin pair. So that's 4 pieces of DNA coming together. And we typically represent that by drawing these things together. They form a particular type of structure. This is just the first stage of synapsis, or the first stage of prophase 1. So, synapsis is the joining together of replicated pairs of homologous chromosomes. Sorry, let's do this a second. You want to take a picture? Or if you need anything before I sort of cover it again, go ahead. Yeah, until the screen goes. Or until after class, you can look at it again. Unless I walk underneath it and start raising things. I can go to the other side. We're going to take our homologous chromosomes that have been replicated. So each chromosome is a sister chromatin pair. And those two homologs come together in a process called synapsis. And synapsis is going to form the structures that we call tetrads, which are the 4 pieces of DNA linked together. Now we draw them as these X's. I wish I had another board so I could draw that at the end of May or something, but sorry for everybody on this row. While we draw them like this as a simplistic view, in actuality, they should be drawn like this. That is a more appropriate representation of what a tetrad actually looks like inside of a cell. Can you see all pieces of DNA? And that's sort of the point, is they're really close to each other, but they're also right on top of each other. It's not side by side. It's basically sandwiched together. One piece of bread, other piece of bread, stacked together. But it's just a little bit easier to look at it as this sort of 4 pieces of DNA. So we can understand and appreciate that it is a tetrad. And there is an orientation there that's important. So we snap, join the DNA together, form the tetrad. If synapsis doesn't occur, crossing over can't occur. The only way for crossing over to happen is to pull the DNA together first. If you don't pull the DNA together, then you can't swap things from each other. Gotta be close to each other so we can swap things. So as a basic picture, here's sort of another idea of what we should sort of think about. Whenever we see DNA, I always use letters. That usually means I use words. So I like to think about words that only have 1 letter used each time. It's not an inexhaustible list of words that only have 1 letter used once. But our town has that name, so it's good. Martin. Is it Martin or Martin? Where do you put the syllable? Between the R and the T, between the T and the I. Who says Mart? Martin. Martin. Or is it Martin? Martin. Martin. It's gonna be in your head the rest of the day. Anyways, we have homologous chromosomes, because again, how do we define homologous chromosomes? Same gene content, same gene order, same length, same centromere position. They both have M-A-R-T-I-N, exact same order. That's what makes them homologous. We take our homologous chromosomes and we duplicate them, therefore there are now 2 copies of the red one and 2 copies of the blue one that are completely identical to each other. Again, this all happens before prophase even starts. So right here is the beginning of prophase 1. Synapsis occurs, the tetrad has now been formed. These individual pieces of DNA now all come together to form the structure that we call the tetrad. And again, it's easy to see it sideways as 4 different things, but imagine the blue was right on top of the red. That's a better approximation of what it actually looks like. They're not side by side, but like this, not like this, but like that. And because they're like that, they can switch things. Things can go like synapses, forming the tetrad, replicated pairs of homologous chromosomes joining together in a tight knit structure. There's a bit more to the story we'll talk about later. But it'll be a mitosis story. So it really is not important. So synapses brings the DNA into close proximity to each other, and then crossing over is possible. Remember, crossing over is a rare event. It's not always going to occur, but it is thought to happen at least once along the entire length of each chromosome. Some chromosomes are really big, so you could have more than one crossing over event, but at least one crossing over event for every chromosome. That means part of your maternal chromosome gets moved to a paternal chromosome and vice versa. You physically swap DNA from one chromosome to another chromosome. Crossing over specifically means the DNA exchange happens during prophase 1 of meiosis. A crossing over is just an example of what we call recombination, or DNA recombination, or what we call homologous DNA recombination, which is a general phenomenon that happens lots of times outside of the world of meiosis. Recombination, homologous recombination is a general term. A crossing over is the specific name given to the recombination that happens during meiosis. There is a specific location in the structure that gets formed to allow for the crossing over to occur. Its name is there, but I'm not going to ask you to memorize it because I'm nice. It's called a chiasmata, which is still a me again. So while you will know my name for one thing, not for the other thing. And by the way, my middle initial is A, so it's Matt A. Still works out. Just don't key, that's okay. Interestingly enough, when I went to a conference with my graduate professor, we were told to submit our names, and I just put, M.A.Pritchett, because that's what you write your full name, you put your initials. Somebody misread that, so my name was Ma. So they call me Ma Pritchett. Which I guess was fairly fitting, because I was kind of the mom in the lab, making sure everything's in the right place and putting things up and saying, hey, I'm actually doing it right. Like a professor. So Ma works too. You can call me Ma if you want to. Ma Pritchett. It'll be all a little joke. But in recombination or crossing over, we're going to see double-stranded DNA from one chromosome switch with double-stranded DNA on another chromosome. And without a picture, kind of lost, so we'll see a picture in a second. But again, crossing over is sort of rare, so one will happen not very often, two will happen less often, three will happen less often. But the cool thing about crossing over is now we are mixing and matching traits, phenotypes, that were on mom's DNA versus dad's DNA. And so if, let's say, eye color and hair color are on chromosome 1, without crossing over, you would have both your mom's eye color and your mom's hair color. Because that's the way things work. They would always be inherited together. Or you got dad's chromosomes. You get dad's DNA, dad's hair color, and dad's eye color, but never mixing and matching. Crossing over allows that mixing and matching to occur. So we get greater phenotypic variation because we can unlink pieces of things that are on the same chromosome. This is what we call an intra-chromosomal change, because we're only affecting either chromosome 1 or chromosome 2 or chromosome 3. We're just taking the genes in chromosome 1 and shuffling them back a little bit, just mixing and matching those things. But we can't mix and match them all, we can only sort of do parts at a time. But it's only a single chromosome affected when we look at crossing over. We can take the maternal and the paternal and sort of switch them a little bit. And this is one way in which we can represent the crossing over process. So we typically always see there is a crossing over event, which is not to say this is a sister chromatid, but this is our sort of X drawing. And given there's 4 pieces of DNA, we only need 2 for crossing over to work. That's typically what we do see. We don't see all 4 pieces of DNA involved in crossing over, but it could happen. It's just going to be more rare. And so when we look at crossing over, typically look at them crossing over affecting the inner two rather than the outer two. So this one is unchanged, is what we see here. The other one is also unchanged. But in the middle, two, we do see a switch. So now part red, part blue, part red. They both have the same gene content and the same gene order still, but the alleles get swapped. Now crossing over really matters, or only really matters, in a heterozygous world. If you're homozygous, does it matter? I mean, there are different types of recessive alleles, different types of dominant alleles maybe. Maybe one recessive allele is more likely to cause breast cancer than another recessive allele, so maybe then it does matter. But in the grand scheme of things, when you're homozygous, crossing over really is not going to change a whole lot. You're still going to have something dominant, something recessive, or vice versa. And this is just showing you one crossing over event. You could have two occur. Like if you had another one happen down here, you would go red, blue, then back to red. And that's another possibility of things that could occur. We're just trying to swap DNA. And because we're swapping DNA, we're going to get a lot more variety of allele combinations together. Now, luckily for Gregor Mendel, he was lucky. Remember, Gregor Mendel said that everything was inherited individually and separately. That's the idea of independent assortment was the phenotype of the trait controlling the seed color is inherited separately than every other trait they look at. If it's on the same chromosome, the only way to reconcile that is if crossing over happens. But Gregor Mendel didn't know about crossing over. So he got really lucky. But the genes he was looking at, or the traits he was looking at, were either really far away or on different chromosomes. And I'll show you later that he's lucky. Because some of the things he looked at were on the exact same chromosome. And the only way to make sure that they would actually be separatable is in a world of crossing over. Something we'll try to talk more about as we go forward.
So when you talk about intrachromosomal, it's within one chromosome pair? Like
Between the maternal and the paternal copy
Okay.
of the same chromosome.
Okay.
Yes. And it's not, the idea of intrachromosomal versus interchromosomal is more about the variety that we're creating. We're not dealing with chromosome 1 versus chromosome 2, which is the next thing. We're dealing with chromosome 1. So that's what I call it intrachromosomal, because chromosome 1 maternal, chromosome 1 paternal can switch things up, but it's still chromosome 1. But crossing over allows us to separate, or potentially separate, alleles and traits that are on the same chromosome. It's the most important thing about crossing over. It allows us to separate the traits that are on the same chromosome. You can get some different combinations of phenotypes that were not present in either parent. Especially for things that are on the same chromosome, because that's what we care about. Crossing over allows for the unlinking of things that are on the same chromosome. At least the potential unlinking of things on the same chromosome. There are some caveats to this process. There are some things that can go wrong. This slide just mentioned one, but we'll come back to this one in a second. The idea of translocation. But there are sometimes mistakes that are made during crossing over. And as we're almost out of time, we'll have to wait until Wednesday to hear about these possible problems that can occur.